Dear students, are you preparing for NEET 2023? If yes, then it is a good news for you all that CareerPoint is coming with its new course for class 11 students, where we are providing live interactive classes. Live interactive classes by experienced and topmost faculty team of Kota. A student can ask their doubt during lectures using live chat. Class notes are provided in the form of PDF for every class. Live lectures are also provided in recorded form. DPPs, daily practice problem sheet, are also provided in PDF form and recorded form. Exercise sheet along with solution provided to all the students in PDF form. Printed study material package and exercise sheet and live exercise sheet with discussion classes. Doubt solving counters. Two-way interactive mode, video conferencing based doubt counters by subject mentors. Online test series along with the feedback. This includes chapter test, unit test, part syllabus test and full syllabus test. We not just provide test series, we also provide classes for the discussion of test papers. So live classes for paper discussion. Last but not the least, motivational seminar by our experts, individual mentoring and parents-teacher meet. All this will be conducted on eCareerPoint learning app. For more detail, visit our website eCareerPoint.com. For any query, call us on 0744 6630 We are here to make your future.
Download eCareerPoint app. Best live online quota coaching at your home.
Hello, hi, a very good afternoon. So this is Krishnaveni and we've been talking about the story of life on earth, and that is biology. Yes, hello FF, very good afternoon. Okay, so I understand you have written something in Malayalam, but I'm sorry I cannot read Malayalam. So please can you translate it for all of us? Yes, hello, hi Binu. So very happy good afternoon. So Satyendra Singh, hello, hi, good afternoon. So Anthony Abhi, hi, good afternoon. So Harini, good afternoon. So Harini, thank you. So thank you everyone for joining my session. So please do like, share and subscribe. So for all those who are new to my session, so Monday, Wednesday and Friday I teach for class 12 and Tuesday, Thursday, Saturday I teach for class 11. So, okay, so there's someone new as well to our session. So, hello, so that is Prajwal. Hello, yes, I am good. Thank you so much. So, thank you so much, Prajwal, for joining our session. So, please do like, share and subscribe. All right. So, here we are talking about your excretory substance and their elimination. So, basically, we are talking about your excretory substance. So, we are towards the last bit. So, one small portion is remaining for us because last class we were talking much in detail with a lot of interest about the mechanism of concentration of filtrate, right? So, I was not able to finish this chapter. So, today we'll talk about, so we were also done with mutation if you remember. So, I will just do the last portion of disorders and also the other organs that are used in excretion. All right. Yes. Okay. So let's move forward. So this is what we were talking. So we were almost done. So we were almost done with the regulation as well, if you remember. So we were done with mutation also. So only what is remaining is your disorders, right? So don't worry, I will just give a recap for all those who are new. So we started off this chapter. So this chapter has a weightage of 2% in your entire human physiology. But the most tricky thing is this chapter because they can confuse you with a lot of things, because one is the mechanism of formation of urine and then it is the mechanism of concentration of your filtrate. So what is the storyline of this chapter? So let me tell you. So first we start off talking about the nitrogenous waste. So your nitrogenous waste are your ammonia, urea and your uric acid, right? Yes. So how does the human body excrete these nitrogenous waste and what are the methods, right? So based on the type of nitrogenous waste, the organisms were classified as your ureotelic. So the organism whose waste product is urea, then uricotelic, Then amenotelic, right? So these were the three ways in which we classified organisms and according to their excretory waste. Then we also started talking about the excretory structures that is present from the lower phyla, that is from your platyhelminthes, we went forward. So in platyhelminthes, it was your flame cells or your nephridia, right? So then we went on to see that earthworms have your platyhelminthes, yes, it is flame cells and protonephridia. Earthworms have nephridia, and your ascihelminthes have excretory tube, and your annelids have malpighian tubules. So that is what we were seeing. So the next thing that we spoke about is your excretory structure. Then we went on to talk about the structure of your kidney, that is your human excretory system. So do you remember the structure of kidney? Okay, you are asking if I am from Kerala. No, I am not from Kerala because I cannot read Malayalam. All right? Yes. So you're saying lots of love from Kerala. So thank you so much. So this is me sending lots of love from Kota. All right? Okay, fine. Okay, so if you use emojis, so the night bolt will send you a warning. So reduce the use of smileys. All right? Okay, fine. Yes, green gland crustaceans. Yes, you're right, FF. Okay, so structure of kidney. Your kidney is bean-shaped structure. It is located between your third lumbar and your last thoracic vertebrae. So you're on the concave side of your kidney. You have a opening called as hilum. So inner to the hilum, you have your funnel-shaped structures known as renal pelvis, which has projections on top, which is known as calyces, right? So the outer layer of your kidney is surrounded by a tough capsule. Yes. And your kidney, the inner surface of your kidney is divided into medulla and cortex region. So the medulla opens into medullary pyramids, and it also has projections known as calyces, right? Then in your cortex region runs around the medullary pyramid, and it is known as columns of Burney. So these were the two things that we saw. Then your kidney has the structural and functional unit, which is called as nephron. So we have almost 300 billion nephrons in each of your kidney. So your nephron has two parts. So what are the structures of your nephron? So your nephron has two parts, right? So one is glomerulus and the other one is your renal tubules. So these together make up your nephron, which is the structural and functional unit of your kidney. So what is glomerulus? Glomerulus is a tuft of capillaries. So tuft means a network of capillaries. So capillaries means blood vessels, right? So if I say it is a tuft of capillaries, so someone has to supply blood into the glomerulus and someone has to take blood out from the glomerulus, right? So the one that supplies the glomerulus with blood is your afferent arteriole. Fine. The one which drains out blood from your glomerulus is your efferent arteriole. And glomerulus is always enclosed by a cup-shaped capsule, which is known as Bowman's capsule. So this is your glomerulus, this is your Bowman's capsule. So your Bowman's capsule plus your glomerulus is called as Malpighian tubules or renal corpuscles. Fine. So glomerulus is done. So the next part of your nephron is your renal tubule. So from your Bowman's capsule, it continues to form a highly convoluted structure known as PCT. So PCT further comes down to form your hairpin-shaped loop that is called as loop of Henle. Then you have a mildly convoluted structure called as distal convoluted structure. And finally, this opens out into your collecting duct. Right? Your collecting duct further opens out into your renal pelvis via your renal medulla. Right? So this is the structure of your renal tubule. So this is nothing but your loop of Henle. So this first limb is your ascending limb because it is coming down. Okay? Then this side it is moving up. So it is the ascending limb. Alright? Only bio class in this channel. No, you also have NEET Hindi medium channel. So this is English medium channel. So you also have NEET Hindi medium channel. So there also you have bio classes happening. So two other faculties are teaching. I guess Rohit sir and Shejal ma'am. So both of them are teaching there. So English medium channel I am teaching. All right. Okay, so क्या हुआ? So afferent is one which brings in, efferent is one which takes out. Okay, so no confusion. All right, fine. So this is the descending loop, this is the ascending loop of Henle. Fine, this is the structure of your kidney. So your kidney has two portions, right? Cortex region and your medullary region. So your PCT, your DCT, and what is here? Here it is your Malpighian tubule, right? So all these lie in the cortex portion of your kidney. Fine. So sometimes the loop of Henle is also too short to extend into the medullary region. So in that case, your loop of Henle is present in your cortex region itself. So it is called as corticular nephrons. Fine. So when the loop of Henle extends into the medullary region, it is known as juxtaglomerular nephrons. Alright, is this clear? Okay, so this was the structure of kidney that we saw. So one last point to add. So your afferent arteriole forms a network of capillaries around your renal tubule. So these are your renal tubule, right? So your afferent arteriole forms a network of capillaries around your renal tubule. So that is known as peritubular capillaries. So this peritubular capillaries, they form a minute blood vessel very parallel to your loop of Henle, and that is known as vasa recta. So very parallel to your loop of Henle, you have a minute blood vessel, that is your vasa recta. Alright, is this much clear to everyone? No doubts? So this is the structure of kidney. So after we were done with the structure of kidney, we went on to talk about the mechanism of urine formation. So we had 3 steps in your urine formation, right? So what are they? So the first one is your glomerular filtration. The next one is your reabsorption. And the third one is secretion. Right? So these are the 3 steps of your urine formation. So the first one is glomerular filtration. So filtration of blood by glomerulus is known as glomerular filtration. So the blood from the ventricle, that is 1/5 of the blood from your ventricle, reaches your kidney, especially into the glomerulus via your afferent arteriole. So your glomerulus is a network of capillaries, and basically it is going to filter blood. So what are the 3 layers involved in the filtration process? The first one is the epithelial cells of your glomerulus. Since the glomerulus is a network of blood vessels, the epithelium lining your blood vessel is known as endothelium. So it is the endothelium of your glomerulus, epithelial cells of your Bowman's capsule, because your glomerulus is always enclosed by a cup-shaped capsule known as Bowman's capsule. Epithelial cells of your Bowman's capsule, which are known as podocytes. And in between your glomerulus and your Bowman's capsule, you have a basement membrane. So your filtration process is going to happen between these 3 layers. Alright? Yes? So these podocytes have filtration pores which help in filtering. So this process of filtration is so pure. So what it is filtering? Blood. So what are the constituents of blood? Plasma and formed elements. Of course, your formed elements which has your cells will be eliminated. Your plasma which contains your plasma proteins. Even your plasma proteins will not be filtered. Only your plasma will pass through this filtration process. Yes, you're absolutely right. So it is known as ultrafiltration process. Alright? Yes? Okay, so the amount of blood filtered by the glomerulus per minute is known as glomerular filtration rate, and the value is 125 ml per day and 125 liters per minute and 180 liter per day. Right? So if at all there is a fall in your GFR rate, what gets activated? Your JGA cells get activated. So these JGA cells are present at the junction between your distal convoluted tubule and your afferent arteriole. Alright? So these JGA cells in turn activate your renin. Yes? So this renin will activate your angiotensinogen to angiotensin 1 and angiotensin 2. Alright. Then what will happen? So if the pressure is less, which means the volume is more of the blood vessel. So if you have a less GFR rate, your JGA is activated, which in turn now will decrease the volume. So this angiotensinogen will act as a vasoconstrictor and bring back your GFR rates. Alright, fine. So now if you are if your glomerulus is filtering 180 liters of blood and you excrete only 1 to 1.5 liters of urine per day, so where is the remaining 99% going? So the remaining 99% is getting reabsorbed by your renal tubule. Yes, so you have your PCT, DCT, loop of Henle collecting that. So everywhere it is getting reabsorbed. Yes, you are right, FF. Fine. Okay. So your water is reabsorbed by simple diffusion process. Your nitrogenous waste also by simple diffusion process. Your sodium, sodium, glucose, amino acids are reabsorbed by active transport. So active transport requires energy. Alright. And the last step is secretion. Since you are reabsorbing a lot of things from the filtrate, to maintain the ionic balance, you also secrete some ions. Ions like your H+, ammonia and K+, right? So this is the mechanism of urine formation. Clear? Okay. So the next thing that we saw was the function of the tubules, right? So what was the function of each of the tubule? So the first tubule is your PCT. So what happens in your PCT? So if PCT is where maximum reabsorption takes place and it is lined by brush border epithelial cells. So one more point, your PCT has increased surface area because that is the region where maximum reabsorption occurs. Clear? Is this much clear? Okay. So PCT absorbs maximum essential nutrients as well as 70 to 80% of your electrolytes, and it is also involved in the secretion of your H+, K+ ammonia, which helps in maintaining the ionic balance. Alright, and it also secretes bicarbonate ions into the filtrate. So this is the function of your PCT. Next we were talking about your loop of Henle. So the loop that comes down is known as your descending lymph or the descending loop. The one that goes down is your ascending loop. Right? So the upper portion of your descending loop that is pretty thin, yeah, so that is lined by your cuboidal epithelium and the lower region is lined by your squamous epithelium. Alright? So your descending loop of Henle is permeable to water and it is impermeable to electrolytes. So, before your loop of Henle, you have your PCT. So, a lot of ions, water are getting absorbed in your PCT. But as it is coming down, it is coming down into the medullary region, which is more osmolarically active. That's which means it has less concentration of water. So more water is present inside the renal tubule. That's why by the process of diffusion, water moves out from your descending lymph since the membrane is permeable to water. So that's as the filtrate moves down, it is getting more concentrated here. Alright? Yes? Okay. Which is more coiled? PCT is more coiled. Okay? Yes, why are you typing it so many times? Yes, ascending loop of Henle dilutes the urine. No, it is not. Yes, you're right. Ascending lymph dilutes because it is exactly opposite to this. So this lower portion is lined by squamous epithelium and upper portion is lined by cuboidal epithelium. Okay? Yes? Fine. So your ascending loop of Henle, since it is permeable to electrolytes, your NaCl moves out and whatever amount is present inside, it is getting retained. Okay, so that your filtrate is diluted here. Then your filtrate moves into DCT. So DCT helps in conditional reabsorption of your water. of your sodium. Alright, and it also helps in the secretion, that is H plus, K plus and ammonia into the filtrate. The last one is your collecting duct. Your collecting duct extends from your cortex to the medullary region, alright, and it also helps in the reabsorption of water, and it also has a small passage for the reabsorption of urea. So these are the functions of your 4 tubules, okay. So you want me to teach photosynthesis? Alright. After physiology or human physiology only, I can start plant physiology. So in between I cannot take, right? Yes. So once your entire human physiology is done, maybe then I will start off with photosynthesis. Alright? Okay. Okay, so I revise every day, so it is easy. Yes, that is the main psychological reason to revise, so that it stays in your mind. Fine. So the next thing that we were talking about is the mechanism of concentration of filtrate. So when your body is highly dehydrated, when you lose water by sweating, when you're having diarrhea, so when all your ionic balance is getting imbalanced, all the water is getting lost, so that time your kidney has to conserve water, right? So the mechanism by which your kidney conserves water is known as countercurrent mechanism, and that is the mechanism by which it is concentrating the filtrate. Done? Yes? Okay. So one more announcement that I forgot to tell you. So tomorrow you might have a mentee, and the mentee will be at 6:30 if it is there. Okay. So and the topic for the mentee that I have not decided. So which topic you want for mentee? Any idea? Yes, any idea? So shall we have body fluids and circulation? So we were done with digestion, breathing. So tomorrow it will be body fluids and circulation. So that will be the topic for mentee. All right? Yes? You want to have cell cycle for mentee? It's been a long time. Okay, we can also have cell cycle, no issues. Yes, others, you want to have ecosystem? Yes, ecosystem is also done. Okay, so last I had organism and population, right? So this time we'll have a class 11 chapter only. So class 11 chapter, do you want cell cycle or do you want body fluids and circulation? Okay, so I will have 3 mentees this week. So yes, I know cell cycle is difficult. So let me give you a revision of cell cycle before I take a mentee. Alright, so tomorrow I'll have body fluids and circulation. Then one extra lecture I will take. So maybe in the evening on Thursday or Friday, I will have an extra lecture where I will revise your cell cycle, and then the following Friday or Saturday we'll have a mentee on your cell cycle. Alright, yes. Hello, hi Priya. So how are you? Doing good? So thank you so much for joining my lecture. So please do like, share and subscribe. Alright, so that will be the agenda. So tomorrow if I have a mentee, so I will be having it in your body fluids and circulation. Then I will take an extra lecture on Thursday evening maybe. So where I will revise cell cycle, and then I'll have a mentee on Saturday. Alright, is that okay, FF? Fine. So how does the mechanism of concentration of filtrate occurs? So it occurs between your vasa recta and your loop of Henle. So what does these two do? So inside your vasa recta you have the blood flowing, right? Arey, kya hoga? Uh-oh. Yes. Okay, so inside your vasa recta you have your blood flowing, and inside your Henle's loop you have your filtrate flowing, right? So in your Henle's loop, this is the descending lymph and this is the ascending lymph. So in your vasa recta, it is exactly opposite. This is the descending lymph, this is the ascending lymph. This will exactly fit like this. So in your ascending lymph, when your NaCl comes out, this in turn is caught by your descending lymph of your vasa recta. So once again, your vasa recta moves into the interstitial fluid. So this is the medullary portion, right? So that is the interstitial fluid which is getting more concentrated. Why? Because what we have to conserve water. So water should not get excreted in the filtrate. So that is why when more electrolytes come, then water will automatically move out, right? Yes. So more solute molecules, then the water will move to a solute. So in the interstitial, yes, now you're having lot of electrolytes. So that is the idea. There should not be any water. So this is point number 1. Then I told you, your collecting duct also helps in reabsorption of urea. So your collecting duct once again concentrates urea in your interstitial fluid. So once again your filtrate is getting concentrated and water is not getting excreted here, meaning your urine is not getting diluted here. So this is the mechanism of concentration of filtrate. Then we spoke about the regulation mechanism, how hypothalamus regulates with the help of osmoreceptors, right? So how it activates your pituitary to release your ADH, which in turn acts on your DCT, which performs the role of conditional reabsorption, right? Then your JG cells do know renin-angiotensin mechanism, yeah. So it will act as a vasoconstrictor, and it will also act on your adrenal gland to produce your aldosterone, which also helps in the absorption of water. Fine. And heart produces ANF. So ANF decreases your blood pressure, meaning ANF acts as your vasodilator. So do you remember this hormonal mechanism? Yes, by which your kidney functions are regulated. So is this clear? No doubt? Yes. So shall I go into the next part? So I guess exactly this is okay. So mutation is the next part. So we were actually done with mutation. So a quick recap. So the process of release of urine is your mutation, right? So the neural mechanism that is causing it is your mutation reflex. So what happens? Your filtrate, once it comes out of your nephron, is stored as urine in your urinary bladder, right? So when urinary bladder is full, the stretch receptors of your urinary bladder send a signal to your CNS. Your CNS will in turn send a signal which says, okay, yes, it is ready to be excreted, because it is under the voluntary control, right? So once the CNS sends back the information to your urinary bladder, then your ureteral sphincters relax and the urine is excreted. So that process is known as mutation. Fine? Don't worry, it will be completed. Okay? Vasoconstrictor and diluter. Constrict is to reduce the space. Dilator to dilate, to increase the diameter. Alright? Yes? So don't worry. Constrictor to constrain. You constrain your efforts, right? You limit your efforts, meaning it is becoming small. So dilator, it is when it is increasing the diameter of your blood vessel. So this is mutation reflex. So here we also spoke about that your urine has a pH of around 6.8 and it is slightly yellowish in color. So it has a characteristic order, and you eliminate 25 to almost 30 grams of urea in your urine. So if your ketone comes, okay, so which is vasoconstrictor, which is vasodilator? Okay, so when your GFR is less, your angiotensin gets activated. So when your GFR is less, your volume is high, right? So now we are trying to decrease the volume. So angiotensinogen is a vasoconstrictor. ANF is a vasodilator. Alright? ANF is a dilator, angiotensin is a constrictor. So 20 to 30 grams of urea is getting excreted, and if glucose is excreted via urine, that is glycosouria, yes, so which leads to your diabetes condition. So this is your mutation. So exactly we were here. What are the other organs for excretion? So we have 3 other organs: lungs, liver, and of course the last one is the biggest organ, that is skin. Okay, so lungs. So what is the role of lungs? Lungs helps in the exchange of gas, right, between your carbon dioxide and oxygen. So lungs excrete a major chunk of carbon dioxide. So lungs also plays a role in excretion. So the liver secretes bile. So bile has bile pigments, bile salts. It also reabsorbs some of your drugs and your minerals. So that also helps in your excretion process. And finally your skin. Your skin has 2 types of glands. Okay, so one type is your sweat gland. The other one is your sebaceous gland. So sebaceous gland are oil and wax secreting gland. Sweat gland produces your sweat, right? So your sweat is a watery sodium containing fluid which when evaporated produces a cooling surface on your skin. So via your sweat also some amount of junk is getting excreted. Then your sebaceous gland. So your sebaceous gland is an oil secreting gland, and through this also some of your substances are getting excreted. So when I say other excretory organs, I want you to remember three things: lungs, liver and kidney. Sorry, lungs, liver and skin has two glands, sweat gland and sebaceous gland. So sweat gland is the gland that produces your sweat, and sebaceous gland is your oil gland, and it also produces wax. Okay, so this is beneath your skin. Both are present in your skin only. So this oil producing gland, it also eliminates your microorganisms. So it is responsible for producing moisture or oil on your skin. So otherwise your skin becomes very extremely dry, right? So when it is extremely dry, then your pores are not very effective enough, and it can let microorganisms pass through, right? So that is why it should always be in an oily state, so that it is easy to swipe off the microorganism. Yes, keratinized. Yes, so keratin is a protein, so that is also present. Fine. Okay, so the last part of your excretory system that I want to teach is your disorders. So we have a handful of disorders. The first is uremia. So malfunctioning of kidney. So if your kidney malfunctions, then you have accumulation of urea in blood, right? So that is the problem. So that leads to uremia. So where does urea come from? So urea is an excretory waste, right? So we excrete urea 25 to 30 grams in a urine. So urea is being formed in your liver, and it comes to the kidney for excretion. So that is uremia. So when the kidney does not function properly, urea gets accumulated. So how it is being what is the therapy? What is the diagnostic method? That is hemodialysis. So I will teach you the entire method of hemodialysis. Hold on in the next slide. Hare, diarrhea, ingestion, vomiting is for digestion. Yes, so we are talking about the disorders of your excretory system. Then acute, so when this condition of your uremia continues, then it goes to acute renal failure, okay, for a long time, right? So that is acute renal failure, and then the only solution can be transplantation, kidney transplantation. Actually on Saturday I forgot to tell you, so Saturday was the 6th of March, right? So 6th of March is considered as the World Kidney Day, sorry, the World Kidney Month. So where it is open for donation of your kidney. So so if you're willing to donate your kidney, so you can register on that site. So there is a national site. So I guess it is not world, it is National Kidney Month. So that was celebrated on Saturday. So when I was teaching, so before class I remembered that I have to tell you, but during class I forgot. Okay. So just type dialysis, and there is one page which is your which the government of India has created, so to create awareness. So because it is not always possible to have kidney transplant, right? So you should have a compatible donor. So that is why they say please donate kidney, because in today's present scenario there are a lot of kidney failure cases, and if a person with two kidney fails, then the person is going to die. It's going to be fatal, right? Can we remove that poison before it reaches the blood? No, definitely not. I will tell you why. Because, so what is the blood that comes from your, what blood comes to your kidney? The blood that is being pumped by your heart, the one that comes out of aorta, which goes into your lower abdomen, comes inside your kidney. So you have separate artery that goes into your brain, right? You cannot stop that. Only 1/5 of the blood that is being pumped by your ventricle is coming down into your kidney. So the remaining 4/5, you have no control, right? So that is the problem. Yes, you can live with one kidney, but that one kidney will get enlarged when it is the only functional one. All right? Okay. So continuous uremia condition leads to acute renal failure, and the only solution is transplantation. The next disease is your renal calculi, when you have stones or insoluble mass of crystallized salts getting accumulated in your renal tubules. Okay. So you say kidney stones, right? They are your renal calculi. So kidney stones are really painful. So please drink a lot of water. So glomerulonephritis, whenever it ends in nephritis, it means inflammation, swelling and reddening of that particular portion. So inflammation of your glomerulus. So when you have inflammation of your glomerulus, which means your glomerulus is not able to perform ultrafiltration effectively, right? Okay. So this is your hemodialysis. So hemodialysis is the method of artificially excreting urea. So what happens is, so where do you take out blood from? So what blood comes to your kidney? The blood from your ventricle, right? So basically you have to catch hold of an artery because artery catches your oxygenated blood. Yes? Yes, various kidney are present, but according to an individual, it is not compatible. That is why the rate of acute kidney failure, acute renal failures are very high. Alright? Yes, I agree. I don't deny. So you have a lot of artificial kidney present, but not for a sustained period, not for a long period. For 1 year long, you cannot take. For 6 months itself is a very saturating period. Okay. So you pick up an artery from a person because an artery carries oxygenated blood. So you pick up the artery and connect it to what is known as a dialyzing machine. So this dialyzing machine has a dialyzing fluid. Yes. So your blood passes into the fluid and whatever has to be excreted moves out and then the blood moves inside your veins. Okay. Yes. So one more point, when you take blood out of the blood vessel, it will clot, right? So you have to add anticoagulant factor before you take out the blood. One more thing, so this dialyzing fluid has some concentration similar to your plasma because only when there is a gradient, a concentration gradient, then it is easy to eliminate, right? So this has a concentration similar to plasma, similar to your one that is present in your plasma. So here it is covered by your cellophane tape. It's not cellulose, cellophane tape. So this tape has minute particles. So your blood has urea as well, right? So it has urea, it has nitrogenous waste. So now once it enters inside this dialyzing fluid, it has pores through which your urea, your creatinine and all the other substance that has to be excreted to come out and the remaining constituents of the blood flows back. Okay? Yes, the remaining constant of the blood flows back. So this dialyzing fluid has a concentration similar to your plasma. So your blood enters inside. So it has a cellophane tape. So that has a membrane. So that has pores. So the pores are created such that all your substance that has to be excreted, right, like your urea, creatinine, will pass into the membrane and remain in this remain in this machine itself. So the remaining portion of the blood will flow out and move into your body. So before putting inside your body, you have to take out the anticoagulant here. Alright. So this is the method of hemodialysis. So remember, your dialyzing fluid does not have urea. It has your plasma concentration, but it does not have urea. So your dialyzing fluid is free of urea and urea comes from the blood. Fine. Yes, Shilpa. Hi, hello. So I guess you're new to my session. Thank you so much for joining my session. So please do like, share and subscribe. Alright. So this is all about disorders. So now I have a set of 10 questions. If you guys are quick, so 1 minute for each question. Okay. So will you guys give me the answer as quick as possible? So which of the following statement is incorrect? So I want an incorrect statement here. So in ureotelic organism, ammonia is not a product of metabolism. So in mammals, some amount of urea may be retained in kidney matrix to maintain osmolarity. So in fish, kidneys do not play any significant role in the removal of ammonium ions. Urea and uric acid are less toxic than ammonia. So what is the answer here? Which is used as an anticoagulant? Heparin is used as an anticoagulant. Alright? Yes, what is the answer now? Quick! 1 minute for each question. So I have 10 questions. So if you want to finish quickly, C is incorrect. Incorrect statement. So first one is right. Yes, Shilpa is right. So why it is A? No, it is not C. It is A. Read the question A. In ureotelic organism. So ureotelic organism is the organism in which urea is the excretory product. So the excretory product can be urea, but it does not mean that ammonia is not byproduct of metabolism. Ammonia, by the byproduct of metabolism, is converted into urea in your liver. So this statement is wrong. Okay, in your fishes, Yes, kidney does not play a significant role because fishes do not have kidneys, right? So they are lower vertebrates. So how will they have kidneys? So this is a right statement. So they are asking for incorrect statement. So read the question. Ammonia is not a product of metabolism. Ammonia is a product of metabolism, and then your ammonia is getting converted into urea. Alright. So this is where you guys will make mistakes. So here your answer is A. Fine. So question number 2, columns of Burtney in the kidneys of mammals are formed as an extension of what? So what, okay, we want to note it. Yes, please note down. So column of Burtney in kidney. So column of Burtney is your cortex region. They are formed as extensions of, it is understood, right? So columns of Burtney is your cortex region running around your medullary pyramids. So your answer is cortex in medulla. So your columns of Burtney run around your renal tubule. So it is your cortex in medulla. Alright? Yes, you guys have understood the logic. Shall I go forward? Yes, so you can type the answer once and not so many times. Okay, then Nightbot will warn you. Yes, good. So question number 3, the dotted appearance of the cortex of kidney is due to. The cortex portion of the kidney has dotted appearance due to which of the following? Yes, what is the answer here? The dotted appearance of the cortex region of kidney. So what are the regions present in your cortex? Loop of Henle is not necessarily present in your cortex region. So it is because of your convoluted parts, that is your PCT and DCT, you have dotted appearance in your kidney. So here your answer is B. Okay. Yes, that is because of your convoluted parts. Alright. So shall we go to question number 4? No, Anthony, it is because of the convoluted parts, because it is highly coiled, right? So under the microscope, it looks like a dotted structure. Fine? No, it is not ducts of Bellini. So that does not give a dotted appearance. In your cortex region also, you have your PCT, DCT. It is not only a column of Burtney, right? Okay, question number 4. Liquid which collects in the cavity of Bowman's capsule is? So your glomerulus filters, right? So the filtrate consists of what? So that is what it is asking. Which collects in the cavity of Bowman's capsule. So you have your glomerulus and then you have your Bowman's capsule. So from your glomerulus, your filtrate comes in. So what are the components of your filtrate? What are the components of your filtrate? So it is plasma minus blood proteins and blood cells. Yes. So only your plasma, pure plasma without plasma protein comes out, right? Yes, it is not A, it is here. Concentrated urine does not come out of your Bowman's capsule. That is after it passes through your loop of Henle only. It is not A. Shilpa, no, it is not A. For concentration of urine, you need loop of Henle and you need your vasa recta. Okay, it does not happen in your Bowman's capsule. Okay. Yes, of course, plasma passes, but without your plasma proteins. Yes, ma'am, a very happy good afternoon. So, thank you so much for joining my session. So, question number 5, the net pressure gradient that causes the fluid to filter out glomeruli into the capsule. So, when blood gushes out inside your glomerulus, there is some pressure that is created in the walls of your glomerulus that causes the filtration to happen immediately. So that is known as net pressure gradient. So what is that net pressure gradient that causes the fluid to filter outside the glomerulus into the capsule? So what is it? So here, I don't know if you know the answer, it is 10 mmHg. So please remember this, it is 10 mmHg, not 75, it is 10. Okay? Done? Yes? So shall I go forward? Fine. So question number 6, which of the following option has the correct pair of nephron parts that maintain pH and ionic balance of blood? So, one which helps in tubular secretion, that is the one that helps maintain the pH and ionic balance of the blood, right? Yeah, that is the pressure on the walls, but I am asking about the net pressure gradient. So net pressure gradient comprises of your hydrostatic pressure, the pressure caused by the water that is present, as well as by the blood colloidal particles, as well as by the glomerular wall. So everything put together is your 10 mmHg. Okay. So which of the following helps in maintaining the ionic and the fluid balance? Yes. So which is the answer here? So it is pretty confusing, right? So people say A. So loop of Henle does not help in secretion. No, loop of Henle. So did I ever mention while talking about the function of loop of Henle, it secretes your H plus K plus ammonia? No, I have told you it is PCT, DCT and collecting duct, right? But hold on. So your PCT and Henle's loop, this option is wrong. Henle's loop does not secrete H plus, Na plus and K plus. So DCT and glomerulus also does not secrete. So once again it is wrong. CD and Henle's loop, so this is also wrong. So the only option that is right is your DCT and collecting duct, because both secrete H plus, K plus and ammonia ions into the filtrate. Alright? Yes? So here your answer is B. You got it? How it is B? So if the options are confusing, so try eliminating each one of them. Okay? But here we are talking about maintaining the pH and the ionic balance of blood. So which maintains the pH and ionic balance of blood? So this is your second step of your urine formation. So what is the second step of urine formation? So the third step is your tubular secretion, right? To maintain the ionic and osmotic balance, you secrete something into the filtrate. So your loop of Henle is different. Loop of Henle works in concentrating your urine. So to maintain your pH and ionic balance, it is your renal tubules, alright? So without your loop of Henle, fine? Yes, you're absolutely right, Priya. So that is 10 mmHg, fine? Yes, it is getting out, but not because to maintain the pH and ionic balance. So that is a different case. This is a different case. So what helps in the secretion process during urine? So that is your pH and ionic balance. You're not understanding? So in the mechanism of formation of urine, you have 3 steps. First one is your glomerular filtration, then you have reabsorption. So when you reabsorb a lot from the filtrate, the ionic balance is disturbed. So to maintain the ionic balance, you have some secretion. The secretion is done by your PCT, DCT and your collecting duct. So what do they do? They secrete your H plus, ammonia and K plus into the filtrate to maintain the pH balance. Okay, so what happens in your ascending loop? Ascending loop pulls out Na plus because it is moving into the cortex region. That is by the process of diffusion, not to maintain your osmolality. Alright, so this helps in maintaining the pH. So it is your PCT, DCT and collecting duct. Since there is no option which puts all the 3, so we are logically eliminating the option. Alright. Yes. So moving on to question number 7. A person who is not taking food or beverage will have dash in his urine. Will have dash in his urine. So little glucose, less urea, excess urea, little fat. So basically a person who is fasting. Okay. So when he is excreting, so what will happen? He will have less urea because your metabolic process that breaks down urea will get less of energy. Yes. And since you're not taking any beverages, your body has to conserve urine. So at the time of conserving urine, your kidney has a lot of pressure on itself. So it is basically less urea because there is no energy. Your body is trying to use the energy that is already present. Alright. So it is less urea, not excess urea. So ideally you're supposed to excrete, so 25 to 30 grams, so that is the normal level. So this is when your metabolism is proper. But when a person is fasting or starving, so the metabolism is not ekdam proper, so it is less urea, fine? Yes, you're right. But here, according to the option, we have to justify. So question number 8, which of the following is not excreted normally in urine? Uric acid, hemoglobin, ketone bodies, hippuric acid. So what is the answer here? What is the answer here? So ideally, we excrete 25 to 30 grams of urea in our urine, right? Yes, normal condition. But when a person is not eating, then the body has to conserve its energy. So it will not spend much energy in breaking down your ammonia into urine. So when ammonia is not broken down into urine, so how will your kidney excrete urine, right? So that is the reason it will have less urea. So which one of the following is normally excreted? Yes, you're right. It is hemoglobin. Hemoglobin is not excreted because your glomerulus cannot filter your RBC. So question number 9, the yellow color of urine is due to the presence of urea, uric acid, urochrome and bilirubin. So which one is the answer here? The yellow color of urine. So that is because of which of the following? No, bilirubin is present in your bile. It's your bile pigment, right? So your answer is urochrome. Okay, so that is a pigment present. So chrome is color. So it's not uro, it is urochrome. So this is responsible for imparting the yellow color to your urine. Alright. So question number 10, the last one, dialyzing unit, that is your artificial kidney, contains a fluid which is almost same as plasma except that it has. So your plasma will contain urine also. Yes, your plasma is the liquid portion. It also has organic matter and inorganic matter, right? Yes. So your plasma has urea, but your dialyzing unit will not have any urea. So that is the difference. Okay. Bilirubin and bilirubirin gives color to your fecal matter, not to your urine. Okay, Shilpa. So here it is no urea in your dialyzing fluid. All right. Is that clear? Yes. So this is my last slide for the evening. So that is all. If you genuinely understood this chapter properly, because I have extended it to 4 lectures. So please do like, share and subscribe. So the next class 11 topic will be taught on Thursday. So Thursday we will start off with your locomotion and movement. All right. So meanwhile, tomorrow at 3 p.m. so we will talk about your environmental issues, and we will also have a Menti tomorrow at 6:30 in your body fluids and circulation. All right. On Thursday, I will take an extra lecture where we will revise your cell cycle. Fine. Yes. So thank you for being a wonderful audience. So I will meet you tomorrow. So until then, stay tuned and take care. So bye-bye.